Hey everyone, so today I'm going to be talking about cannabinoid exposure in rat adolescents and how it reprograms the initial behavioral, molecular, and epigenetic response to cocaine. So the initial exposure to an addictive substance can facilitate repeated use. That is, that individuals experiencing more positive effects are more likely to use that drug again. And increasing evidence suggests that psychoactive cannabinoid use in adolescents enhances the behavioral effects of cocaine. However, despite the behavioral data, there's no neurobiological evidence demonstrating that cannabinoids can also alter the brain's initial molecular and epigenetic response to cocaine. The endocannabinoid system has a modulatory role in brain reward and cognitive processes, and it's been hypothesized that repeated interference with endocannabinoid signaling can remodel the adolescent brain and make it respond differently to more addictive substances, such as cocaine. In the present study, we demonstrate that a history of synthetic cannabinoid exposure in adolescent animals results in distinct molecular and epigenetic changes following initial exposure to cocaine. These changes were pronounced in the prefrontal cortex and associated with an enhanced response to cocaine stimulatory effects. The prefrontal cortex is a brain region that still undergoes maturation in adolescence, and its dysfunction contributes to the development of addictions. So for our methods and materials, first and foremost, the animals and behavioral assessments. So we used male Sprague Dolly rats, which were acclimated for 1 week before starting treatment with a synthetic cannabinoid, WIN, cocaine, or a vehicle. We used a series of analyses, namely next-generation sequencing-based analyses, subcellular fractionations and protein extractions, global quantitative proteomics by mass spectrometry and Western blotting, and gene knockout and overexpression. For statistical analyses, we did 2 group comparisons using two-tailed unpaired student t-tests, and 4 group comparisons using ANOVAs. So for our results section, we can actually see the study design is laid out in of figure 1 up here, where the starting day is the 44th day for our adolescent group, the 42nd prenatal day, and the 77th day for our adult group. So this is the actual, so there's the WIN group and a vehicle group, and there are 4 different IP challenges within that. So there's saline, cocaine. So there's 2 groups per each of those groups, and those make up the 4 groups of the study in total. We can also see from this first graph here that the effect was present in adolescents, but not in adults, where there was a significant value observed for the WIN cocaine group over and above the WIN saline and other groups by a significant measure. So there's also some other really interesting information that we can make out here from our other figures. So figure 2, for instance, we can see the observed difference between cocaine versus control as opposed to WIN cocaine versus cocaine, where there has in fact been a noted difference between those, also in terms of frequencies. It is also worth highlighting that there is a decrease in the HDAC6 observed, however, only in the adolescent population and not in the adult population. There's a few things that we can discern from these figures, but most importantly, we find that in adolescents, but not in adult rats, pre-exposure to WIN results in cross-sensitization to cocaine, which correlates with histone hyperacetylation and decreased levels of HDAC6 in the prefrontal cortex. In the prefrontal cortex, we also find that synthetic cannabinoid WIN pre-exposure blunts the typicom RNA response to cocaine and instead results in alternative splicing and chromatin accessibility of events involving genes such as NPAS2. Moreover, pre-exposure to WIN enhances the effects of cocaine on protein phosphorylation, like Gefrin, for instance, which we can actually see down here in figure 5. and modulates the synaptic composition both in the PFC, the prefrontal cortex, and the nucleus accumbens. PFC and NACC gene network topological analyses following cocaine exposure reveal distinct top nodes in the when pre-exposed group, which include PACAP and ADY's AP1. These preclinical data demonstrate that adolescent cannabinoid exposure reprograms the initial behavioral, molecular, and epigenetic response to cocaine.